So it's my great pleasure to introduce the second keynote speaker of today, Robert Plenge, who is the Executive Vice President and Chief Research Officer at Bristol Myers Squibb. He came to Bristol Myers Squibb from Celgene that was acquired by BMS, and before that he was Head of Translational Medicine and Vice President at Merck. He began his journey in academic medicine, training for his MD and PhD on the West Coast at UCSF and UCSD, and then he came to the East Coast to do a fellowship in clinical rheumatology, and he liked the climate so much that he ended up staying here, I guess. And this is also, of course, both clinical rheumatology and genetics, of course, and not just at the Brigham and Women's Hospital, but at the Broad Institute as well. So I think your mentor here was David Altshuler, is that correct? I couldn't quite remember that piece. And he also has brought this unique voice to our community through his blog that I really recommend that you guys read, called Plenge Gen. If you haven't read it already, it's about impact in genetics and medicine, and really has some interesting thoughts about how we should approach drug development through those types of approaches. And I know that this is also what his presentation will be about. And a fun fact, which for those of you who know me, I love fun facts. I actually had the good fortune of interacting with Robert when he was at the Broad. And when he transitioned to Merck, gave me an opportunity to get my first industry collaboration and grant that actually launched my lab. It was my second grant ever, I think around 2012 or 13. And it was a real game changer for me. And I don't think I ever told you that, but it was really great interacting with your team at Merck, and the funding was fantastic. We actually did some really good work with that money. So it's just an enormous pleasure to welcome you back. I've been excited to hear your talk, and I love the title, so can't wait to see what you're going to tell us.
I did not know that, so thank you, and I'm glad we were able to help. So first of all, thanks for inviting me, and for all of you who are still here, thanks for hanging in there. I know it's been a long day. So I'm going to start in the same way that Ben actually started, which is to thank a number of people who actually contributed to the talk. So Joe Moranville, who's part of Bristol Myers Squibb, I've worked with for a number of years. He contributed a lot of the data that I'll show you today. We had a great pleasure to work with a company called Octant. Sri Koshuri was a postdoc with George Church, so kind of starting in the beginning and the end of the day, and they did a lot of the work on saturation mutagenesis that I'll show you at the end of the talk. And then lastly, I'm going to give you a number of examples that have come out of our Bristol Myers Squibb pipeline. I have worked on the minority of them until recently. So there are a lot of people who have worked on these programs for a decade or more, and I'll be able to share some of the examples with you today. So this is a talk I haven't given before. So you're going to have to tell me if this concept of $1 billion bet actually works. I'm going to start in the way that Melina actually started the day as well, which is to frame the concept of some of the challenges that we face in drug discovery and development, which is we spent all of this time doing what Vijay talked about on MSI2 and trying to understand the genetics and the functions of a particular protein, and we do it in animal models, we make molecules, we take them into human clinical trials, and the vast majority of those things just fail. And they fail because the drugs don't work. Somehow we picked the wrong target. So what you can actually see on the left-hand side of this graph is that simple pie chart of why most drugs fail. Most drugs fail because the drugs don't work, despite many, many years of hard work leading up to making the medicine and testing it in humans. And I think on the right-hand side, this sort of frames at a high level, one of the reasons that drugs don't work is we just don't have enough insight into human biology to make all of the right choices that are required to identify a target, make a molecule, take it into the right patient population, test it, and to see this PK-PD relationship and ultimately get a drug approved. So hopefully as we can focus more and more, not just on human model systems, but causal human model systems, we'll begin to improve the rates of efficacy in clinical trials. So I think we've heard about this a bunch already today, and I'm sure you're all very familiar with this, is that human genetics is a very powerful tool to help predict the success of a drug target in clinical trials. So Matt Nelson had a paper a number of years ago. Eric Minical expanded on this a couple of years ago. And basically the take home here is if a drug is against a validated genetic target, it's at least a 2 to 3-fold increased likelihood of probability of success in clinical development. We've done some work on this. Joe Moranville and his team have done some work on this within BMS. And one thing in particular is we've added on this kind of proteoMR approach, which if you can actually have proteomic data linked with genetic data, that's also a good predictor of drug success. But as I'll go through the talk, one of the things that I really want to emphasize is, yes, genetics is a powerful predictor of success for drugs, but there are properties of human genetics which can actually be extended into other areas that we call causal human biology that also can be powerful predictors of success of clinical trials. And so we want to take those principles in human genetics, apply them in other areas. So Chloe talked about some of the work in profiling immune cells. You know, if we can identify those pathogenic immune cells, for example, that are driving diseases like lupus and figure out how to selectively deplete those cells, that's causal human biology. Potentially that is another way to actually come up with new drug targets. Okay, so we've tried to distill some of these very complex topics for drug discovery and development into 5 principles. And behind each of these principles, there's a tremendous amount of information. And one of the nice things about going last in the day is you can kind of pull on the examples over the course of the day to kind of really emphasize these concepts. So when I made the move from academics to industry, it was really because of that first principle. I thought human genetics could change the way that we do drug discovery and development. If only we worked on the right targets and used human genetics to pick those targets, everything else would fall into place. I think the biggest education that I've had since joining industry is I realized that, yes, picking the right target is really important, but there are a lot of other things that come after it that are really important as well. And again, VJ gave a great example of MSI2 to show that how much time goes into understanding any individual target. You don't want to just understand, in this case, a genetic association, but what are the details of the biology of the particular target? How do you want to modulate it? What are you going to measure once you get into the clinic? That's all captured by that single concept of causal human biology. But it's a really, really important starting point for drug discovery and development. So the second step then is to match a therapeutic modality to a molecular mechanism of action. And Ben just gave you a great example of how to actually do that in the form of CRISPR-Cas9. If you understand a mutation that is causing the disease and it's expressed in the liver, you can figure out how to give that drug into the liver and correct it in the liver and make the mutant base go back to a wild type base. That's a perfect example of matching modality to mechanism. We also heard about manipulating BCL11A, and there it's actually not introducing something new, but it's taking something out, preventing the binding site of a transcription factor that allows the repression of fetal hemoglobin. So by removing that site, that's matching molecular mechanism. You're basically recapitulating human genetics to turn on, in this case, fetal hemoglobin that's shown to be a very protective and effective way to help patients with sickle cell anemia. But it's a third category that I've been spending more time on recently. So if you pick the right target, you match the right modality to a molecular mechanism of action, then you take that molecule into humans for the first time. And this is when you really begin to get at dose, how much of the drug is required, schedule, how often do you give it? Do you give it once a day, twice a day, once a week? What's the frequency with which you give the drug? And then what's the relationship between pharmacokinetics, the level of drug in the body, and pharmacodynamics, what that drug is doing to the target or the cells within the body? And then how does that relate to clinical efficacy? So we call this path to clinical proof of concept. And as I'll talk about in just a few minutes, this is the point, really for the first time, that you know if a molecule, a medicine is working as you actually predicted. And so it's this really important inflection point in a drug discovery journey. Then once you see clinical proof of concept, step number 4 is to accelerate clinical development, as many indications as you can and as fast as you can, because there's incredible competition to make not just the first drug, but also the best drug. And there are a lot of people that are doing this across the entire world, and being first and best is incredibly important. And then all along the way, what you want to do is the 5th principle, which is to build out a complete compendium of information that will allow this drug to be used in the most number of patients so the most number of patients that can benefit. So that's maximizing market access. So these are the 5 principles. And so what I thought I would try to do today is to describe this in a way that uses poker as a metaphor. So just before I I start this metaphor. How many people actually play poker? All right, so a decent number. So Texas Hold'em. So this is going to be based upon the game Texas Hold'em. And I'll describe the game to you. And again, this is the first time I've actually used this metaphor. So you'll have to tell me if it actually works or not. So causal human biology, matching modality mechanism. If you play Texas Hold'em, each person is given 2 cards. Those cards are private to you. So I'm playing against Jesse. I'm playing against Ben. I shouldn't pick statisticians because they're really good at this stuff. But I'm playing against them. I've got my 2 cards. They've got their 2 cards. And I can see mine, but they can't see my cards. And in this case, I've got 2. I've got an ace and a queen, 2 pretty good high cards. So this is the target and matching modality to mechanism. So I'm feeling pretty good about the hand. In this case, the hand that I'm taking from research into early development. Any ideas on what the average cost of a drug to take from research into early development is? About how much do you think that costs per drug taken into early development? Any estimates? Anybody want to shout out a number? 100 million? That's actually right. Yeah, 100 million. Wow, this is a great crowd. Yes, about $100 million to make that medicine and take it into humans for the first time. So you have to be very, very careful about how many of these medicines you take into early development. All right, so then you start to do your early development study. This is where you're doing dose and schedule, you're giving it to the right patient populations, and this is when you see a really big inflection point. So at the start of a clinical trial, until the end, it's about a 10% probability of success. At this point, you go from about a 10% probability of success. If you do the right trial, you're going to about a 20, 30, 40, potentially even a 50% probability of success after what's called, at least in poker, the flop. So now you get 3 cards. So Jesse and Ben, there are 3 cards that we all can play together. I've got my 2 private cards. They have their 2 private cards. And then we have 3 in the middle. This is the flop. I'm feeling pretty good because I now have a pair of queens in addition to ace high. This is a pretty good hand. So basically what I'm trying to say here, I hope this metaphor is working, is that in early development, I'm feeling really good at this point. I picked the right target. I've actually matched a modality to a molecular mechanism of action, and I've done this early clinical development study, and it looks really positive. But now I need to complete the full hand. We do accelerated full development, and we start to see, in this case, an ace. Now I got a pair of aces, 2 queens, 2 pair. That's a good hand. But at the end of the day, it's about having a great label the FDA can approve. And in this case, a queen, that's a full house. That hand wins almost every single time. So basically, you need all of these together in order to have a winning hand, in which case this is an approved drug. I mentioned about $100 million to take a drug into humans for the first time, and it's over $2.5, $3 billion to ultimately get a drug approved. But it's not the cost of the successes, it's the cost of the failures that really drives up that price. So this is why human genetics can help us with these billion-dollar bets. So what I'm going to do now is I'm going to go through a few examples of programs in our pipeline, and I'm going to go through them relatively quickly. But the points are really going to emphasize everything that I've just talked about in the last few minutes. Picking the right targets, matching modality and mechanism, seeing that early clinical data, and ultimately doing what you can to accelerate full development. Now, and just one other example here, you could have a competitor that goes after the exact same targets. They pick the right target, they make the right molecule, but they stumble in early clinical development. They don't design the right early development trial. They don't have the right biomarkers. They don't have VJ on their team to really understand deep details of the mechanism and how to understand if this drug is working the way that it should. And so ultimately, they're not successful. So again, it's more than just picking the right targets and making the right molecules. There's a lot that happens in early development as well. Okay, so the first story that I'm going to tell you very quickly is a story that I worked on when I was back in academics. And this was understanding the genetic basis of a molecule called TYK2. And what this is showing is the causal human biology for this is very compelling. There are at least 2 or 3 different common alleles that are loss of function and protect from risk of multiple autoimmune diseases, including psoriasis, lupus, and rheumatoid arthritis. There was a study that was published by Lars Fugger and colleagues, Callie Dendrew, that showed that 1 copy leads to about a 10 to 20% reduction in TYK2 signaling, but 2 copies, that is in the homozygous state, leads to about a 70% reduction in TYK2 signaling. So it's loss of function at about a 70% level that protects from autoimmunity. And then this was one of the first studies that actually did a PheWAS to look for PheWAS as a way to predict adverse drug events. And what we saw with those same variants is they did not increase risk of infection or other blood dyscrasias. So it seemed like not only was it a good target based upon efficacy, but it was a potentially a safe target as well. But it was really hard to make a therapeutic molecule against this target. On the left-hand side is the regulatory domain. That's where that triangle is. There are a lot of other molecules like TYK2, JAK1, JAK2, JAK3 that have a lot of homology in that orthosteric domain. So every single time a company tried to make a binder against the orthosteric pocket, they hit JAK1, JAK2, and JAK3, and that was associated with side effects. So what BMS chemists did, what BMS biologists did, is they designed a phenotypic screen, and they uncovered this allosteric pocket. Actually, I got it backwards. The active site is where most of the homology is. The regulatory domain, where the triangle is, that's where we identified, the BMS scientists, a drug could bind to this allosteric pocket, but it could lock the active domain in the inactive state. And so BMS discovered a small molecule that really recapitulated human genetics in a very faithful way. And then in the first in-human clinical trial, in this case, looking at an interferon signature in psoriasis, you can see about a 70% reduction. So really precisely phenocopying the human genetics. So about 70% inhibition for human genetics and about 70% inhibition for the drug. So there's really nice phenocopy of the drug on this particular phenotype. And so TYK2 is now approved in psoriasis, and we have ongoing trials in other autoimmune diseases, specifically lupus and Sjogren's. So really, if you go through the metaphor, you have full house. This is a successful drug and an approved drug, and we have other ongoing trials as well. Okay, so now I want to go to that second principle, matching modality to mechanism. And to me, again, if I go back to where I was about 15 or 20 years ago in academics, I was really focused on the target. And then I came to appreciate how much there is now in our therapeutic armamentarium. In the late 90s, basically 90% of drugs were small molecules. There were a handful of more conventional antibodies. Fast forward to today, you've got, and this is just the BMS portfolio, you've got a bunch of different ways to modulate therapeutic targets, and there are a lot on this list that we just don't pursue, including many of the genetic-based approaches that you've heard about over the course of the day. But what I'll do is I'll walk through a couple of stories, starting with small molecules, but I'll touch on a few of these. So here is a genetic target, TLR7. There's very strong human genetics as shown on the far left. So TLR7 gain-of-function mutations lead to a lupus-like phenotype, suggesting that inhibiting TLR7 could be therapeutically beneficial. There are some challenges in actually inhibiting this target, and you had to get it, a small molecule, to actually inhibit this within the context of the endosome. And on the far right is that first in human clinical study where we also saw a change in an interferon signature for a drug called afimatorin. This drug is now has just finished phase 2 and is entering into phase 3 clinical trials. And this is some of the clinical data that shows a very nice separation in a subset of patients with lupus that are called cutaneous lupus, so CLE. And what you're actually seeing in the graph in the bottom row in the purple is a nice difference between a clinical score in patients with cutaneous lupus compared to those same patients who received a placebo. Factor XIa, this has been a highly validated target, a genetic target for a number of years. Patients who have factor XIa deficiency are at a rate of mild deficiency in terms of hemophilia. But what I can show you there on the left is the hemophilia is relatively mild, that they're not at a major risk for spontaneous bleeding. They also have a lower risk of VTE. So this suggests that if you can actually create an inhibitor to factor XIa, you can protect from venous thromboembolism without a risk of bleeding. This has been one of the major challenges with anticoagulants, is anytime you actually inhibit part of the coagulation cascade, yes, you decrease the risk of forming thrombus, which can occur in the heart, which can occur in the brain, but that occurs with a bleeding risk, which could be potentially quite devastating. So very strong human genetics. BMS scientists made a small molecule against activated factor XIa And we have human clinical data in a phase 1, phase 2 study, which actually has shown that at least to date, this has a profile that's actually quite compelling. So the purple bars are showing a decrease in venous thromboembolism, and this can be done in what's called a total knee replacement study, but it's basically a decrease in the risk of forming that thrombus, that clot. And then the green bars at the very bottom are showing evidence of major bleed. So what you're seeing is a dose-dependent decrease in thrombus formation in purple without an increased risk of bleeding. This study is now, this molecule is now in 3 different phase 3 trials and more than probably 30 or 40,000 patients across a number of different clinical settings. And we should have data towards the end of next year and early 2027. So we know that the phase 1, phase 2 data are compelling, but ultimately we'll have to wait until the phase 3 data to know if it'll be a successful drug or not. I want to spend just a few minutes on a different therapeutic modality, targeted protein degradation. This is a way to actually not just inhibit a target of interest, but to degrade that target of interest. So the idea here is there are a lot of validated targets, a lot of human disease-associated targets, but most of them are not amenable to more conventional small and large molecules. In the case of certain types of proteins, such as transcription factors or those with scaffolding functions, there's no enzymatic function to inhibit. You have to actually degrade or get rid of the target, which is often difficult to do. And there's also benefit in terms of using protein degraders to overcome resistance of small molecule kinase inhibitors as well. So this is a story that actually builds nicely on the story that Vijay told in terms of being able to induce fetal hemoglobin. What I'm showing you on this slide is that we actually have identified two targets that influence fetal hemoglobin production. One of those two is a genetically validated target. It goes by the name Pokemon, also known as ZBTB7A. The other target is Wiz. And what we found is that we can identify a molecular glue molecule that can bind to an E3 ligase on one part. So it's basically one molecule that's a therapeutic molecule. It binds to an E3 ligase, and then it recruits these 2 transcription factors to a single complex. It shuttles them to the proteasome and leads to degradation. So we can basically degrade these 2 transcription factors, turning on fetal hemoglobin in our preclinical models. And we are in clinical trials today. And I think by the end of the year, early next year, we should know the extent to which this turns on fetal hemoglobin. So if it does work, it has the potential to be an oral small molecule pill that achieves the same level of efficacy as Casgevy, which is an ex vivo gene editing approach. Here's another example where the target BCL6 is a well-known oncogenic driver. Here, the genetic data is not from germline genetic data, but it's from somatic cell genetic data. So there's a lot of great somatic cell genetic data in oncology. Again, you heard about it from Vijay in terms of a clonal hematopoiesis, but many of these oncogenic drivers can actually lead to cancers, and BCL6 is a known oncogenic driver. But like many transcription factors and other targets, it's just very difficult to drug with a typical kinase inhibitor. So what scientists at BMS did is they created what's called a heterobifunctional degrader. So one arm binds to BCL6, the other arm binds to an E3 ligase called cereblon. It brings the complex together, leads to BCL6 degradation. So very strong human biology, in this case from somatic cell genetics. We've got a really interesting degrader that's this heterofunctional biodegrader. And this is also a program that's advancing in clinical development. And we'll know probably within the next year if we're seeing the PK-PD relationship that we expect. Okay, just a few more quick stories before I go on to the saturation mutagenesis part of the talk, and that is biotherapeutics. And here, again, another story of causal human biology. In this case, it's Tregs and cancer. There's no really strong human genetics to implicate CCR8 positive Tregs in cancer, but through single cell sequencing, and you've heard a lot about that today, what we've been able to do is to identify that a subset of Tregs that are identified in patients with lung cancer and other types of cancers are a negative predictor for outcome. That is, the presence of these highly suppressive or highly suppressive CCR8 positive Tregs are a negative predictor of response to checkpoint inhibitors. And so what we predict is by binding to CCR8 on the surface of cells and degrading, basically getting rid of these CCR8 positive Tregs, we'll be able to allow anti-PD-1 and other checkpoint inhibitors to work more effectively. This is also a program that is in first in human studies now, and we should know more in the next year or two whether we've achieved clinical proof of concept. All right, I'm going to go through one more example and do this very quickly because we talked about small molecules and large molecules, but we're also in an age where we can actually engineer living cells to be therapeutics themselves, and this is in the form of autologous CAR T. And then to me, this is one of the most exciting areas of science and biology right now because I used to practice clinical rheumatology. Patients would come in, young women, childbearing age with lupus. They would give them a very strong immunosuppressive, which could get their disease under control. And inevitably, they would ask, well, when do I come off of these medicines? And the answer was almost always never. So imagine being a young, healthy person suddenly getting this new diagnosis and being told you're going to be on immunosuppressants for the rest of your life. It's pretty devastating. So this concept of being able to reset immune memory, to deplete the autoreactive cells, to get rid of them completely, and allowing a new immune system to reconstitute, and basically that new reconstituted immune system is healthy. This is what we're able to do with autologous CAR Ts. So these CAR T cells, but by the way, just on the bottom left, there wasn't a whole lot of genetic data that implicated CD19 specifically. It was a human pharmacology study where an investigator, George Schett, actually took CD19 CAR T, which was approved for lymphoma, tried it in a very small number of patients with lupus, and sure enough, it completely reset their immune system, and they've been off all of immunosuppressive drugs for several years. We've now treated multiple patients, dozens of patients with not just lupus, but other autoimmune diseases, and we've been able to recapitulate this finding as well. So causal human biology is from human pharmacology, matching modality to mechanism. It's an autologous engineered cell that actually kills the CD19 lineage of B cells, and we now have clinical proof of concept, and we're entering into registrational trials. So these are the examples that sort of show how the sort of framework of these 5 principles work in action and how we're using them all the time within our portfolio today. Okay, so I have about 10 minutes left, and I'm going to go through how we're trying to expand our understanding of individual variants to basically build out this toolkit even further. So when I moved from academic to industry, this was kind of my simplistic view of how I thought we could use human genetics to identify new targets. Not just identifying a single allele at a particular gene, but with sequencing, we'd find more and more alleles. And we could say, all right, each one of those X's, that's meant to be allele. That's disease associated. Along the Y-axis is a human phenotype. Imagine high LDL cholesterol, high risk of disease as examples, high body mass index. And if you could find alleles that were associated with a human phenotype that was an appropriate surrogate for drug efficacy, and you could understand the function, gain of function or loss of function, you could begin to estimate these dose response curves, an efficacy curve, and also know if there was an adequate therapeutic window. The challenge has been, and we've been talking about this all day, is we can find that sort of x-axis pretty easily using biobanks. So we can look quantitatively at whether an allele is associated with high or low cholesterol, what level of risk is contributed in terms of risk of autoimmune diseases. But what's incredibly hard is getting that variant to function. Just a really quick story, when I just started my postdoc, Itzik Pierre, I don't know if any of you know Itzik, but we had just done one of the first 100,000 SNP chips from Affymetrix on the HapMap cell lines, and we could actually identify 30% of common variants. And that was a number that totally blew my mind. And I remember Itzik saying to me, and we were sitting over there in One Kendall Square up in one of the conference rooms, he's like, can you imagine if we could just annotate every single variant, not just by where it is in the genome, but by its function? Imagine how much more powerful our genetic association studies would actually be. So not just looking at linkage to sequelarbian and the static, is it associated or not, but you could add a coefficient that tells you something about function. And I remember thinking, wow, it's like, that's a great idea. That sounds really hard. And sure enough, fast forward to where we are today, I think we're getting to a state where we should be able to incorporate these types of weights into our association study. So I'm going to tell you now a story that's going to build on the TIC2 story that I told you about earlier. And so the question here is, can we take a known validated genetic target that has an orthosteric pocket and an allosteric pocket? Can we mutate every single amino acid at this particular protein? And can we uncover the known allosteric and the orthosteric sites where these drugs actually bind? And can we learn new things about this particular molecule that could potentially help with large-scale association study? So just quickly in terms of what we know about the genetic variation across TIC2, and the upper graph is the ClinVar variants. So there are 3 variants, rare variants, that can, when TIC2 is knocked out completely, causes primary immune deficiency. The next graph below is the NOMAD variants, and there are several hundred variants, and that's shown at the top part of the bar graph, and above a particular frequency spectrum is what's shown below. And if you blow this out and look at the frequency spectrum, what you can see on the far right are the 4 common variants. 3 of these are associated with protection from autoimmunity. But if you take a threshold of 10 to the minus 4th or less, there are about 447 misses SNPs from the UK Biobank that are within this particular category. These are all below 0.001 in terms of the minor allele frequency threshold. We know nothing about the function, but can we use saturation immunogenesis to assign function to each of these 447 variants? So this is where we work with Octant to do a deep mutational scan, where again, we mutated every single amino acid by every other possible amino acid across the entire protein. And we did this with 2 different assays. The first assay that I'll tell you quickly about is an interferon signaling assay. What you can actually see is there's a cassette that's inserted that has different TIC2 amino acid changes that codes for the TIC2 that will bind to the interferon signaling receptor. It'll signal down through, and there's an interferon signaling response element, which can then be read out in the form of a barcode. So about 11% of amino acids, when mutated by this assay, could lead to changes in interferon signaling. And you can see in particular this line where a stop variant was introduced. But you can also begin to see domains, and I'll describe these domains in just a minute. The second assay that we did was a protein abundance assay, so a simpler assay. And I'm going to come back to this because I think this has some application as we think about scaling this approach up. But a protein abundance assay where we also introduce every single amino acid variant into TIC2, and then we can sort by GFP also with a barcode and just measure relative protein abundance. So about 20% of amino acids when mutated lead to a signal in this particular assay. And what I think you can appreciate is there's a lot of overlap between the 2 different assays. And when you look at where the allosteric site is located, this is the name of the drug deucravacitinib, you can identify the allosteric binding site with this functional assay. And you can also identify very clearly the catalytic site. And at least in the protein abundance assay, you can identify the disease-associated P1104A variant. Although it's not quite as clear, I know it's difficult to see, it's not quite as clear that variant has an effect on interferon signaling. So there are other positive controls that gave us confidence that the assays were working, including different annotations by alpha missense, some literature-associated loss of function variants, and then 3 variants that were reported as pathogenic. Okay, and I'm going to come back to the 3 start sites because these identify potentially novel allosteric sites. So this identifies, again, a number of variants that could be loss of function or gain of function by protein expression along the y-axis or interferon signaling along the x-axis. As I mentioned, P1104A, which is one of the disease-associated variants, comes up with a good signal by protein expression, but not by interferon signaling. And I think with these types of assays, we can begin to model where these mutations are occurring on the 2 domains of TIC2, the regulatory domain and then the kinase domain. We also refer to these as the catalytic and non-catalytic domains. And you can see again the active site, allosteric site, in the location of P1104A. But there are these variants that look like they have an effect on interferon alpha, but no effect on protein abundance. Could this be a potential way to uncover novel allosteric sites? So what we did is this filtering mechanism to look for variants that affected interferon alpha signaling along the top, whether there was evidence of destabilization. If not, could this be potentially a catalytic allosteric site? And you can see here, we're just annotating a few of these potential sites, which actually 2 of these sites are right at this kinase, pseudokinase interface, as shown by the blue stars. And this is a full-length view of the protein, where you can actually see in yellow here is the kind of this ribbon of where the receptor itself is going through the protein, and the protein is binding around it. And there are potential sites, not just on where the protein binds or interacts with the plasma membrane, but also where TYK2 is engaging with the interferon receptor. Now, one other way that we actually use this data is to actually look at where the drug deucravacitinib binds, and can we actually map residues that are interacting between the gene, the protein TYK2 and the known inhibitor at the allosteric site. And the quick version of this story is, yes, we can, as we actually go through and use different concentrations of the drug. So lower concentration of the drug, you really find only those sites that are directly interacting with the drug itself. And at higher concentrations of the drug, you identify other sites as well. So it provides a nice kind of functional complement to structural data. Okay, so we started by saying if we can only just annotate the human phenotype data by function, and what I've told you, at least for TYK2, we've been able to use deep mutational scan to begin to assign functions to each of those 447 variants. And so now I want to go back to basically the question that Itzik had proposed 20 or so years ago, is if we begin to incorporate this information into our genetic association studies, can we actually have more well-powered rare variant associations? And so we took these variants and we annotated whether not just by common and rare, but by level of TYK2 expression. And we used a couple of traditional burden test methods that are shown here without any weighting, and it didn't find an association with the 35,000 cases from the UK Biobank compared to 308 controls. But we began to incorporate the output from the DMS variant scores into a model. We began to see more statistically significant associations with this genetic burden test. So again, suggesting that as our methods of assigning function to each of the variants become more and more powerful, we should be able to improve power of rare variant association studies. I will say, and listening to the talks today, sort of occurs to me that if we can do this more and more, not just with this types of functional data, but with the predictive data and the modeling data that has been talked about today, hopefully we'll be able to do a better job of basically incorporating these types of weights into our association studies. And in fact, here are a few newer methods that don't use functional data directly, but do appear to have stronger and more well-powered statistical results using the same data set. Okay, so just 2 more slides and then I'm going to stop. I want to then sort of talk about genetic associations and how this is used. We're using this information to guide indication selection. So what I'm showing you on this particular chart is 3 variants within TYK2, and you can see for psoriasis, RA, and lupus, these 3 variants are protective at a very high level of statistical significance. Inflammatory bowel disease has just a different pattern here, where one of the 3 variants looks like it's a risk variant. And this is something that we haven't really been able to figure out, but it does look like it has a very different pattern in inflammatory bowel disease. And at least the clinical trials to date have been successful in psoriasis. They have not been successful in inflammatory bowel disease. That is, so TYK2 did not work in treating patients with inflammatory bowel disease. And as I've mentioned, studies are ongoing with lupus. So we think and hope that it'll work in this particular indication, but we'll know more by the end of the year. But if you actually sort of plot out the different variants in a slightly different way and look at the opposing effects across the proteome. Again, you see in some cases the variants act in the same direction. In other instances, they act in opposite direction. Okay, so I'm going to stop here, but just to conclude, I know I went through things pretty quickly. Most drugs fail because of lack of efficacy. Hopefully, human genetics can help. I talked about placing billion-dollar bets, using these principles to think about making the right decisions, not just for the target and the modality, but how do we actually do those early clinical studies, and ultimately, how do we do full development for drug approvals? Genetic dose response curves can hopefully be used to define exciting targets and this allelic series, and the deep mutational scans in TYK2 have the ability to further build out these genetic dose response curves. And so I guess the question that I would leave you all with is what should be done next? Is there a way for us to do saturation mutagenesis across a larger proportion of the proteome? Or are the computational methods sufficiently strong at this point in time that we don't need to necessarily do these types of functional studies? So there's a lot I think we could certainly discuss. Before I do, I want to let you know that the paper on deep mutational scan should be posted on bioRxiv this week. Here are the authors, and again, a special shout out to the scientists at Octant who did really all of the saturation mutagenesis work, but working closely with Joe Maranville and his team to work through the associations that I described to you. So thank you very much.
Thank you, Robert. Any questions?
Lovely talk, Robert. And I'm very fond of it, too. So
I feel like there's like a but coming here, Ben.
The question I'm going to ask is about the TYK2 functional assay.
Yeah.
I think, to me, a lot of this turns around the quality of the gene-specific assays and how well we think they're actually recapitulating the function that may be perturbed by the variants that we're introducing, right? So you got to really trust the assay part. And so I guess the question is, how many genes do you think we have that caliber of assay for? Is that assay capturing all of the functions for TYK2? And how do you think about approaching it in that way? Versus, I think a lot of what we've heard about today is, transcriptomics and general cellular properties, but they're maybe not specific about the function of the protein itself.
And there was a lot of calibrating in the interferon assay. There was a second assay that actually never worked, measuring IL-23 signaling that we never, were able to sort of validate with the positive controls. P1104A is a known functional variant, and it didn't come up in the interferon assay. I mean, I kind of am intrigued by this protein abundance assay. So maybe that is something that could be done at scale and is sufficiently agnostic to sort of nuances of biology. So that's the one that I sort of wonder, is that something that we could actually scale up and make relationships to circulating proteins as well? Yeah, Brad?
Yeah, this is me? Oh, yeah, nice talk. Remember David Altshuler talking about the arrow of time, taught me about that 15 years ago or something like that. This is a beautiful application. The one exception was the CCR8 or getting rid of the Treg populations. And there's so much data now on changes in populations of cells and microenvironment and cancer and other diseases. That's not quite causal though, right? It's just associated. So how do you're clearly using that in as a premise for one of your studies.
Yeah.
How widely are you using that, and how do you get at causality in those cases?
Yeah, so I think in that case, you're totally right. I mean, you're basically looking at an association, the presence of these Tregs in patients who don't respond to checkpoint blockade. That's a correlation. There's not a causal relation. We try to actually look more longitudinally to understand, what do we know about the phenotype of those Tregs? When do they infiltrate? But at the end of the day, it's more of a correlative relationship than a causal relationship in that specific case. So I think, we often say, causal human biology, yes, no, and it's far more, I think, of a gradient. And I would say something like TYK2, very strong. That's probably a 3 to 4-fold increased probability of success. CCR8, I'd probably put it as a 50, 100%, maybe a 50% increased probability of success, but not the same level as I think some of the genetics that we actually talked about.
But one right here. Very cool talk. And On the TYK2 screen, I was wondering about in the case where you don't have changes in abundance, but you do see effect in activity, and how much of those changes were due to disruption or creation of de novo phosphorylation sites, altering, protein activity, which is a way that a lot of these kinases are actually regulated by, and how much of those actually exist naturally out there, and just.
Yeah, it's a great question. I'm not totally sure. So I don't know. I mean, the way that we kind of frame this is maybe these are novel allosteric sites, but I think that particular biology is not entirely clear to me. So I think that's going to take more work to sort of sort out.
Thank you.
All right, a question in the back there.
Thank you. That was really interesting. I was wondering, when we talk about how genetics informs drug discovery, my impression is it's mostly on the sense of like one variant going to one trait. But as your example in the last few slides showed, we have a lot more, much richer phenotyping of individuals now. And now we have the opportunity to look at variant pleiotropy. Do you have any comments on whether there's something what can we do with pleiotropy now that we have this data? Do you think it will be useful?
I mean, I think pleiotropy can be used for a couple of things. One, it can be used to guide indications as a way to say it works here is going to work in the next 2 or 3 indications as well. But I think pleiotropy can also be used to begin to predict, adverse drug events. And so pleiotropy can work both ways. It can help you with phenotypes that are good surrogates for efficacy, but also help you for good phenotypes that are surrogates for toxicity. I know we're over time, so maybe we can stop here and head over.
Maybe one last. I think there's already a mic out in the audience.
Thank you, Robert. My question is related to your last comment about what's next. Should we do the deep mutational scan, maybe more systematically? So while you're talking about it, that's really remind me in oncology field. I came from oncology field, right? Yeah. That when we discover all the somatic mutations in patient and indicating maybe certain genes is oncogene, and having a deep map where every single gene are knocked out and functional effect measured across thousands of cell lines where we can correlate. So that's really a deep kind of knowledge base we use to build that kind of a functional relationship and confidence of a gene is a causal gene. So in some way, I would think if we can have this kind of systematic functional data, that will really help us to interpret the genetic data in a much more objective way. Yes. I don't know if it's feasible, but I think that would be great to have.
Yeah, I think the more, I mean, we talk about how we train and build these models. I think we often train them off the data sets that we actually have. I sort of imagine a world in which we actually, try to do far more, like say, mutational scans to look at protein abundance, to uncover allosteric sites, and maybe then that could help us feed, the models that we actually use to identify allosteric, these regulatory domains in a way that's it's kind of difficult to do today. But it does come back to some of the assays, as Brad mentioned. So I think in oncology, for example, for cell cycle growth and basically things that accelerate growth or decelerate growth, those are some assays that I think are amenable to this type of approach.
Thank you.
Great. Thank you so much for the presentation, Robert. Please join me in giving him another round of applause.